Welcome, welcome. That needs to be attached to the outside. Five, 4, 3, 2, 1, start. Turn to a partner and tell them how you did. So I'm about to ask you to show me how well you did. But I want you to actually tell me how well you did, not just the number half of you just are used to showing me, right? Show me how you actually did. 1.5? 4? Other than a fit of the giggles? Okay. Just out of curiosity, how many of you, I know the class voted, right, to meditate at the beginning of class, so I know that most of you like the time. I'm curious how many of you actively try each time to meditate. Just out of curiosity. And I'm not going to take it away just because you're not using the time to meditate. If you're using it to like rest or whatever, that's your lookout. But I'm curious how many are actively trying to meditate during that time. A fair number of you? Good. Sometimes yes, sometimes no. Is that what that means? Okay. Sometimes yes, sometimes no. I can't ask for more than that. You may be able to tell I'm kind of losing my voice a little bit. Yeah. So please help me out a little bit as well. I think I can shout if I need to, but I don't want to find out what that would feel like. So let's try to avoid that. Whenever I start to lose my voice, it always gets a little deeper too. So I always wake up in the morning, it's always like, hey. This weird, like, super deep thing that's not my voice, and it freaks me out a little bit. Apparently I talk to myself first thing in the morning. Hey. Oh, look at that. Today, we're going to talk about, specifically first, we're going to become awesome at something called transcription. We're going to watch this video to help solidify that. What's going to happen? Can I have you track me, please? What does track mean, Will? Just to refresh everyone's memory. Not just eyes. What else? Yes, but what else? Eyes and shoulders, right? I want them facing me. That helps me know that you're paying attention. So eyes and shoulders towards me, please. Good. So, the video we're going to watch, it's going to be a lot of information very, very quickly. You will have questions. What should you do if you have a question? Ask it. And what happens in the course of a video, it's easier to kind of finish the video first. So if you can't ask the question immediately, what should you do so you remember your question? What should you all do? You need something to write with, right? Get out something so you can write down any questions. The board works here, or you can get out a computer, a piece of paper. Get out anything you need. No, those are my Expo markers. And one of them is a permanent marker. So, you all have computers, which work great.
I'm sick of computers.
That's fair. Fair.
Is this teaching us one of the assessments?
Well, Mr. H is sick of you.
Okay,
That's
which one is
okay.
it?
Well, we did Punnett Squares last time. We're more focusing on DNA transcription translation. You did? Nice job. Hey, let me hang on. Yeah. Got it right? What was that? Just let me know what the question is, then. You're going to need to speak a little louder. Pavlov. Pavlov is his name. That is a great question. It's a psychological question. And actually, I know a fair bit about it being a teacher. I'll tell you a little bit about it after we're through the video. But I think it's a good question. You ready?
I'm fighting ready.
You ready? Ready? Ready to go? Ready here? Ready?
Yeah, shuffling my
Okay.
workout though.
Ready? Ready? Ready here? Ready? Did you take notes or write anything down? Ready? Peyton? Okay.
Should I use my phone for notes?
As long as that's all you're doing. You ready?
I'm ready.
Ready? Ready?
Just play.
Ready? Ready? What are you going to write your questions down with? You two ready? What are you going to write your questions with? Okay, hurry and go do that.
Protein synthesis.
Okay, who remembers what is the central dogma of biology? I want you to turn to a partner and just try to pull it out there. If you can remember anything about it, turn and tell it to your partner. Central dogma of biology.
Wait, is it the DNA RNA? No, is that
Okay, I know what we're talking about.
Out of curiosity, don't say the answer out loud yet, or not loudly. I want you to raise your hand and give me a 5 to 0. 5 being I know exactly what the central dogma is. 0 being I have no idea what you're talking about. Okay, listen carefully. I'm going to put you on the spot. Is that all right? What is the central dogma of biology?
Like information stored in DNA and DNA passes to ribosomes and the ribosome uses to make it.
Excellent. Do you understand? Turn to a partner. Recite the central dogma.
It's
I want you to show me again. How many of you feel like 5, you fully understand, 0, you have no idea? So I have a lot of 4s, I still have only 1 5. Let me restate it this way. I made you memorize 3 words. DNA, RNA, protein. That is the process by which proteins are made, correct? That is the central dogma. Turn to a partner and recite those 3 words. If you feel like you understand those 3 words, give me a 5. If you feel like you understand none of them, give me a 0, or anywhere in between. Okay. I can take mostly 4s and some 5s in there. That's good.
You should have a standard of all.
Are those real sneezes?
No, I just like sneeze for fun. Kidding, no.
That makes
it
sense.
wasn't.
What'd you say, Blake?
Everyone always asks that.
That's good. That's all we're looking for. So there are 2 types of nucleic acids. DNA, which stores information for long term. RNA, which stores it for a short term. So it's another type of nucleic acid. That's the NA. Just with some minor differences. Okay, so I want you to write down 2 words. These 2 words are going to be the framework. These 2 words are going to be the framework for what we're about to learn, okay? This is something that is easy to mix up if you're not paying attention. If you're paying attention, we'll be good. You all ready? Again, people usually say yes or no in the conversation. Okay.
Yes.
Good? Here's the words. Transcription. Transcription. Are you writing these, Lexi? Transcription. And the other one is translation. The first word you just wrote down, I just want to make sure we understand what this word means. What did you all just do?
Transcribe.
You transcribed. What did you do? What are the actions? What did you do? You wrote down what? What I just said. That is transcription, right? Going from essentially the same language, but just in different forms. When we go from DNA to RNA, have we changed nucleic acids? Yeah, we've changed from deoxyribose nucleic acid to ribonucleic acid, but is it still nucleic acid?
Yeah.
If you will track me for a second. Is it still, Lexi, if you will track me? That means eyes and shoulders. When you transcribe in this process, it's the stepping from the DNA to the RNA. That step is called transcription because you're still nucleic acids. You're not changing languages. We're still talking in English, in other words. Does that make sense? Or am I mixing my metaphors too much? Okay. The next step, want to hazard a guess? What is the next step? If the first step is DNA to RNA,
The protein.
RNA to protein is called translation. What are proteins made out of? Spencer. What are the building blocks of proteins?
You called me Spencer?
Yes, I did. Because I had my friend McCoy. Spencer McCoy was my friend growing up. Lucas.
Wait, say that again.
Three mistakes.
Spencer, you would look at me like, what the-
Yeah, sorry. One of my really good friends growing up was Spencer McCoy. What are the building blocks of protein?
Amino acids.
Good. Excellent. So notice, we shifted from, in the first step, we went from DNA to RNA. They're both nucleic acids. We're speaking the same language, right? On the next step, we're switching from a nucleic acid to what?
A protein.
To amino acids, which will build a protein. We're changing languages. Si estoy hablando así en español, no sé si alguien acá me entiende. ¿Me entiende? ¿Está bien? ¿Me entiende? And if I were to translate what I just said, I said, I'm about to speak in Spanish. Does anyone here understand what I'm saying, right? I'm translating it from one language to another. So that's how I remember, as a mental trick, the difference between transcription and translation. Turn to a partner and tell them the difference between transcription and translation. Back to me in 5, 4, 3, 2, 1. Show me by show of hands, 0 to 5. I want you to show me how confident you feel. 5 being very confident, 0 being not confident. Okay, we're going to throw a lot of words at you coming up in this video. Pay close attention. What do you do if there's something you don't understand? Write it down. You ready?
Yes.
Okay, track the screen. Phrase protein synthesis. Where have you heard the word synthesis before? Photosynthesis. What does photosynthesis mean? So close. But you actually said part of the correct answer. Was that? It makes sugar, right? Uses sunlight to make that sugar. So the word photo means light. What does synthesis mean? Create. Protein synthesis means what? Protein making. You had a question.
With the transcription,
Yes.
is it saying that it makes a copy of itself?
It's going to send... So let me refresh one thing to make sure we're on the same page. If this is a cell, what do we call this organelle? The nucleus, right? What's inside the nucleus? The DNA. Does the DNA ever leave the nucleus? No. So for the information stored in the nucleus to leave, you have to make a copy. And we make a copy using RNA, which is a short-lived kind of break-apart kind of molecule. Its only purpose is to take that message out of the nucleus. Does that make sense? So we are going to take, Ryan, the information and make a copy of the information, which we can then send out of the nucleus. Does that kind of answer your question? Tell me your understanding of... Ask me your question again.
Well, because I thought with the transcription it said that it makes a copy of itself, because with the transcription it's just basically doing what it already was before, it's just duplicating itself.
So we can call it replication when you're making another DNA strand. If we're going to separate the two DNA strands and then make an RNA strand off of one of them that we can then remove and put them back together, so you have that RNA strand that's a copy of the information, that's what we call transcription. So it's not precisely making a copy of itself.
It's just taking a part.
It's making a copy of the information that's in there. Kind of like putting in a thumb drive and copying a file onto it and pulling it away. Because it doesn't change the original file at all. Yes?
How does it get the oxygen into the ribose? Because doesn't DNA not have oxygen?
So DNA specifically, the molecule itself, it's built without a certain oxygen. The RNA sugar, that's the ribose sugar, is just floating around. So when they make that RNA molecule, they just take the one with the oxygen to make the RNA. Good? Okay. What is this organelle called down here? Who said it? Say it louder. That's the ribosome. Okay, what happens at the ribosome? Protein synthesis, which means what again? Creation of the actual protein. Here we go.
In fact, most of DNA doesn't code for anything. only about 2% of it was for protein.
Turn to a partner and summarize what you just learned. Let's see, who's your partner? Pick somebody. You can be with these two.
Some strange. Does the RNA it brings information to a new cell so that they can create one?
It's going to stay inside the cell. It's just going to leave the nucleus because the ribosome is still floating around inside of it.
What's a polymerase?
Polymerase? A polymerase, anything that ends in ASE. Do you remember how we said everything that ends in OSE is a sugar? Anything that ends in ASE is an enzyme, which is a type of protein that does work. So basically all it's saying is it makes a new molecule. Uracil is the replacement for T in RNA.
What is polymerase?
So I'm going to answer it because I think more people have that, because this is the second time I've had that question. So let me answer to the class. So, polymerase. Everyone say polymerase. Polymerase. Does anybody know what a polymer is? If you don't know, I'm just curious. Does anybody know what polymerase is, or is willing to hazard a guess? So remember when we talked about having long chains of molecules with the links that make them up? For example, what's the chains that make up DNA? Or the links that make up DNA? Nucleic acid is the name of the whole molecule. Nucleotides, right? What about protein? What are the building blocks of proteins? Amino acids. Another way of describing this would be to say that DNA and protein are polymers. What does polymer mean? Many. Poly means many. It's made out of many repeating parts. So a monomer would be one of those repeating parts. A polymer is the whole thing. So a polymerase, the ASE ending, means it's something that makes a polymer. So RNA polymerase, what does it make? It makes RNA because it's a polymer. Does that make sense? It's simply an enzyme. It's a protein that goes along and puts everything together. Answer the question?
Yes.
What is RNA polymerase?
So it's the stuff that's in between the links, but it's the opposite, kind of. Because it's like what makes up the polymer. The polymer is like the substance of itself. The polymerase is like all of the individual.
Close. You're close. A polymerase, let me do another analogy. I think this will make it clear. If a polymerase, let's say if a polymer is a chain, okay, and each of those individual chains is called a monomer. The polymerase is the person putting those links together. The machinist or the blacksmith or whoever makes chains. The chainist, I don't know. Whoever puts all those links together and makes a big long chain, that would be the equivalent job of the polymerase. Do you understand?
Yeah.
What do you understand?
It's not the actual links, but it's the thing that's making it.
Yes, it's the worker, shall we say. Does that make sense?
So it makes the DNA and the RNA?
It's the actual machine that's putting the pieces together, yeah. Good? Okay. What are the base pair rules? Let's go, Blake. Give me anything at all in your memory about base pair rules. It can be wrong, that's fine.
So like if one has a base pair, the other has to have the same base pair to like
Very good.
match up.
You're like, I think you're this close to actually having completely the right answer. Okay? So we have how many nucleotides in DNA? 4. What are they? Just letters is fine. A, T, C, and G, right? A pairs with what? T. And C pairs with what? G. Those are the base pair rules.
Apple, whatever.
Yeah, apples grow on trees. Carrots grow in the ground. Do you understand? What are the base pair rules?
So A, T, C, and G have to match up with T.
And who matches up with who?
The DNA.
So like A matches up with what?
T.
And C pairs up with?
G.
Good. Now, RNA does not have T. What does it have instead? U. Okay? So if it doesn't have if it doesn't have T's, it has to necessarily replace that T with the U. So you notice in this strand right here, what would normally match up with this A, Blake? A T would be right here, right? So when I'm doing these so I don't make mistakes, I pretend like the whole thing's DNA, and I write it out just like normal, with T's. But because I know it's RNA, then I go back through and I erase all the T's and I replace them with U's. That just seems like a simpler way to me. Does that make sense? If I were to cover this bottom, would you be able to tell me what the other side should say?
Yeah.
Show me. Write it down.
Wait, say that again?
I want you to write what the pairs are for the RNA. If there's an mRNA strand down here, what pairs with these first 4 bases? Can you write it down, Lexi? You can type it on your computer. If you're there, raise your hand just so I know. Okay. Ready to compare your answers? Did you get it right? Excellent. Okay. Are we good? Are there other questions that have been raised up by this point? We all understanding?
Yeah.
Okay, let's keep going. Questions? Another name for a protein. A polypeptide would be in a completely completed protein.
What does the T mean in tRNA again?
Transfer, because it's transferring the amino acids.
What did you say the codon was again?
So a codon, in DNA, if I look at a 3-letter segment, that's called a triplet. And it's going to eventually code for a specific amino acid. That makes sense, right? What makes sense?
What you just said.
What did I just say?
It said that the codon is like something transferred.
Okay, so DNA holds what kind of information? Gannon?
Doesn't it like code for proteins?
It does. It has information on how to code for those proteins, right? How does it do that? We do it by 3-letter segments. In every 3 letters, it means a specific amino acid, right? So we call that a triplet in DNA. When we're talking about that same 3-letter code that's been, what's the word I use to say when I go from DNA to RNA? Transcription. When I talk about the transcribed triplet, I'm now using a word that represents the same idea, that's codon. So codon is the same thing as a triplet, but in mRNA instead of DNA. Does that answer your question? So what then is an anticodon? Explain. Or you say opposite, you're right, but what does that mean?
Like the opposite of the 3 codons.
So if the codon says AUG, what would be the anticodon? AUG? Yeah, no, you're right. UA, and then last one would be G, and C. Perfect. That's the anticodon. Where do I find the anticodon? Was that? Sometimes. Sometimes not. Look at the picture. Where would I find the anticodon?
Nucleus.
Close.
In the ribosome.
Sometimes. What is the third type of RNA that we haven't talked about much? Somebody actually just asked me what it stands for.
tRNA.
tRNA. The tRNA has the amino acid on the other side, and it has the anticodon on the bottom, and that helps it recognize the correct codon. You following? I want a yes or a no.
I'm a little.
Show me on a 0 to 5. I'm going to actually have you clarify in just a second.
Yeah.
0 is I'm completely lost. 5 is I understand completely. Okay, where are we? What was yours? Okay, where are you a little confused?
Just a lot of it.
Okay, let's do this. I'm going to walk through it on the board using a pen. If you will track to this board at the front of the room, we're going to act as if we are the ultimate controllers of the cell. I'm going to give you a DNA code. I'm going to have you transcribe it. Make sense? We're going to use this code in the future. Let's see.
Transfer.
I want you to transcribe this code. If you don't know what that means, raise your hand, and I'll come help.
What are we transcribing to, DNA or RNA?
The answer's in what I asked you to do. Because I said transcribe, right?
So I don't know what that means.
Okay, so transcribe is going from DNA to RNA.
Okay, so then I know what that means.
Okay. I want you to transcribe this. We're going to do it together. First of all, I want to highlight something. What really matters in this code is every 3 letters, right?
Yes.
These 3 letters, we have a name for it. What is it called? Not yet. It will be called a codon. These are 3 bases in a specific order. Okay, this is good to know. We call this a triplet, which will become a codon. Okay? So in the DNA, every 3 letters, what is it called? Turn and tell a partner. You got it? Okay, so let's go ahead and transcribe this. T pairs with what? A pairs with what? Normally it'd be a T, right? But we're going to RNA, so we're switching to U. What's next? Turn and check your partner's work, see if there was any mistakes, and if you can help them understand. Anything I need to clarify? Okay. Now look back up here. My time again, please, in 5, 4, 3, 2, 1. If you will track front. Eyes and shoulders front. Okay. So, in this, what did we call this? A triplet. What do we call this? Codon. Everyone say codon. Good. Okay. This is, what do we call this molecule? It is RNA. It's a specific type of RNA. It's not the tRNA yet. mRNA, because the M stands for messenger RNA. This molecule is going to leave the nucleus. Where is it going to go? To the in the cytoplasm, to the ribosome, right? The ribosome is then going to read these codons to be able to figure out what amino acids it needs to hook together in what order. Okay? So when this enters into the ribosome, the first codon it sees is AUG. There's a whole bunch of what are called tRNAs. tRNAs essentially are just sticks with amino acids on one end and what we call an anticodon on the other. What would be the anticodon for AUG? Only it's RNA still, right? UAC, right? And this codes for an amino acid known as methionine. I'm just going to write MET. So what all the ribosome is doing is that as this comes in, it just says, oh, that's AUG. I need a tRNA that has the matching anticodon, and I'm going to match them together. And if they match, that's my first amino acid. Make sense? What would the next amino acid be? Or what would the next anticodon be? So then it would, through the flood of all these different, it's like I'm teaching Spanish in here. CCC. Oh. There would be another unique amino acid on the end. And it would say, okay, I need a CCC anticodon. And it's going to look through everything, find one, see if it matches. And if it matches, then it's going to hook those 2 amino acids together. Does that make sense, Ellie? What makes sense?
The fact that even so, like it copies it to their anticodon, and then if and when they match, then they'll hook it together. So then you have a complete set after that was taken over the DNA.
Perfect. I want you to explain to a partner the following. What is the difference between a triplet, a codon, and an anticodon? Discuss. So I'm almost out of time. So this is going to be my last explanation. What is the difference between transcription and translation? Tell a partner.
Makes a copy
Okay, my time again in 5, 4, 3, 2, 1, and track. Track me, please. Do you understand transcription? Yes or no?
Yes.
Do you understand translation?
Yes.
Could you explain this and teach this to a new person?
Yeah.
Could you try?
Yeah, I could try.
And is there a chance that you would succeed?
Yes.
Okay. We're going to finish this video in the last 5 minutes of class or whatever. We'll finish watching this. It's going to go through how we know which codon codes for which amino acid. It'll explain a little bit about how to use those charts. Be prepared to teach this to somebody that doesn't know this. I'm going to try to arrange for you to teach somebody else this process. Does that make sense?
Wait, teach who?
You'll see.
Like someone like that.
Who doesn't know this yet? Yeah, out of this class. That's the goal. We'll see if I get it to work, but be prepared for that. Here's the rest of the video. Like me, going from not DNA, RNA to protein. I want you to translate what my code is, what amino acids would generate, those would generate, in what order. I'm going to go back, there's a chart. Sorry. I'm going to go back just a little bit.
Wait, so are we going to have to know that to be...
You don't have to have memorized the chart, but you need to know the process.
Okay.
Does that make sense?
Yeah, so like we're not gonna know we're not gonna need to know the specific amino acids.
I don't. Yeah. So, up on the board is the chart. Up here is the code. I want you to tell me which amino acids this is calling for.
Wait, which one is the code?
That's part of the question.
No, like on the board, like where is it?
The top strand is the DNA. The strand underneath that is mRNA.
Still on the left side.
What's that?
Is the first one tyrosine?
First one's not tyrosine.
I don't get how to do it.
So the first letter of our one we're looking at is A, right?
Oh, I
Oh, I see
didn't.
the second. Oh.
Yeah, so start with the A. Then what's the next letter? So I'm going to look for the second letter area. It was now a column. So see where the A and the U line up.
So is it methyl?
It's going to be either methionine or isoleucine, right? We don't know yet. But we look like there's a third letter.
Yeah, so it's
G would be methionine.
okay.
Does that make sense? Write these down. What's that?
No. Are we finding the anticodons for
No,
this?
just the actual. You're asking what are these codons asking for?
Okay, so
The anticodon's more just about knowing what that means.
so we're looking for these on that.
That's correct. Yes. A good point was raised. You use the codons for this. This chart is used for the codons, not the anticodons or the triplets. You use codons for this. Where do you find the codons? On what molecule? The mRNA, right? The mRNA molecule has the codons. That's what you use for the chart. You guys, by the way, have been awesome today. Like, truly awesome. My throat has been really painful. You made it easy. You paid attention. You worked hard. I know it's been a long day for you guys, and you still worked your butts off. I appreciate that.